So on this big topic of language, I think it's always good to start with the auditory system, because those of us who are hearing, not talking about sign language here, but those of us who use language as a spoken acoustic medium, we rely on acoustic signals. And those of you, I know there are lots of people here in the audience who study phonetics. This is, of course, very familiar to them. These are so-called spectrograms that we can use to describe, the nature of... So this is frequency plotted over time. So speech is obviously a time-varying signal in the acoustic domain, and you see that there are frequency changes that occur when we speak regular words, and there are harmonic elements in there. You see those regular, sort of equally spaced distances in the frequency domain. That signifies that something is a harmonic sound. We also have noisy parts in our language that are generated by a different part of the vocal apparatus. But they're all pretty simple elements that make up such an overall complex phenomenon like speech. And the question is now, how does the brain process these complex time-varying signals that we have no problem deciphering? We learn to decipher them, to be sure. I have a granddaughter that is just 14 months old, and she's just very interesting in terms of language development. I kind of made these up. I mentioned to Wolfram Ziegler that I made these observations when her father, my older son, was learning language, and unfortunately those observations that I made were in a notebook that got lost, that burned during my moving to the United States. The moving van burned down, and we got all these notes burned down, but I'm trying to do this again now on my granddaughter. So the question is, how does our brain online and in real time process these very complex signals that we are constantly, that you're now hearing, through your ears, and your eardrum is starting to vibrate and making these little vibrations. And how on earth do we do this? I find this an incredibly fascinating question. It's one of the most fascinating questions to me in all of brain research. And it starts with that process, and we only know very little about it. So here's a slide that is taken from Corbina and Brodmann's work, who actually did this work here in Munich, and I also show it in other cities, not just here. It has a special meaning here, of course. He was at the predecessor institution of what is now the Max Planck Institute for Psychiatry, where I did my PhD, and he was one of the first anatomists who made brain sections of a human brain and also of different animal brains to look at fine differences in the cortical areas. The cortex is the highest, most highly developed part of the brain that surrounds our cerebrum, the sort of biggest mass in our brain and phylogenetically the youngest part. And he characterized this purely on the basis of the staining of cells in these areas, and was able to classify them and put numbers on these brain areas. So area 17 at the very back of our brain is the primary visual cortex that is essential for us seeing complex sound, sights and letters and so on. Faces sort of beginning to be processed there. And this was an area that was studied by Nobel Prize winning work of Hubel and Wiesel in the 1960s and 70s, and is still a very, very important model system for understanding the brain, because we understand a lot about vision. Hearing, on the other hand, which happens here, area 41, was named by Brodmann as primary auditory cortex. That's where the sounds arrive after they go into your ear, into the brainstem. This is where the kind of real processing happens and begins. And then it spreads out into surrounding areas, 42, 22, that are also have to do with hearing. And subsequent work in mostly patients that had gunshot wounds, for example, during World War I, and later even starting in the American Civil War, then led to characterization of what these areas are actually doing functionally, and that it turned out that this is for hearing, this is for speech. There's another slide that I took out, but it's kind of Kleist in Germany was one of the first to do all these characterizations. And now we jump, since this is about language, we jump to what are generally known as the language areas in the brain, named after a German neurologist, Karl Wernicke, and a French neurologist, Paul Broca. And both of them found that in people with strokes or with gunshot wounds, for example, when these areas are lesioned or destroyed, then people have impairments in language processing. And they're of a different nature. Broca found that when this part of the frontal cortex is damaged, people have problems uttering sentences. They have agrammatism, so they can't really form sentences. They can still produce sounds and to some extent understand speech. Whereas Wernicke found that if this part, in what we call the temporal lobe or superior temporal cortex, is damaged, then people have so-called sensory aphasia. That's what it used to be called. And people can still speak sentences, but they don't make sense sometimes. So this is how we started in the late 19th century, and now we're trying to work our way up to an understanding of the auditory and language system the same way as people in the visual system have begun to understand how we see faces, for example, how we see letters and so on, and how we read. So the first part of my talk will be about this, about the cellular basis of auditory perception, because I think we have to understand that before we can go to more complex parts of language, and how auditory areas are built in a very similar way to the visual cortex. There is a hierarchy of areas that the signal basically runs through in order to identify more and more complex sounds, and that same principle applies to speech sounds. So speech sounds, in my opinion, are just one category of complex sounds which we use to communicate and which we use to make sense. I hope people here agree with me on this, although I know there's a lot of controversy, and I think it's a parsimonious, simple principle to assume, first of all. So here is the inner ear stretched out. It actually looks like a snail wound up in our inner ear, and different frequencies are represented along this membrane. So at the base of the membrane, which is sort of close to the eardrum, if you wish, so there's the middle ear in between, so I'm sort of simplifying now, there's high frequencies are represented there. And at the apex of that snail, the very end of the basal membrane, low frequencies are represented. You can imagine there's somebody from Hungary, from Decay, actually got his Nobel Prize on figuring out how this worked. There was somebody here in Munich, Eberhard Zwicker, who was one of my professors when I studied here, who also helped to figure this out. So maybe he should have shared the Nobel Prize. But von Decay really came up with the crucial idea that the coding of sound frequencies in the inner ear happens like in a rope. It's called a traveling wave. So if you have a rope that you attach to the other side of the room, you have another wall here, and you then stimulate the rope with a wave. And you do this faster and faster. Then you get actually a location along that rope that resonates with your frequency that you use for stimulation. And you will notice that the faster you do it, the more closer to your action the peak of that vibration will be. So it makes sense from a mechanical point of view that high frequencies are represented here at the base where the membrane is attached, and slow frequencies, when you just do one, then the traveling wave has time to run all the way to the end of the rope. And that's why low frequencies are here. Anyway, so this is called tonotopic representation. Just as in the retina of the eye, you have a retinotopic representation where basically like an electronic camera, you have different parts of the visual field represented at defined places along that receptor sheet. And this is important to understand because everything that is represented here, the receptor cells here, project up into the brain, first the brain stem, nuclear nucleus, and up to the auditory cortex in the same fashion. So we get the same principle of tonotopic representation where frequencies, high frequencies are at one end, and low frequencies are at another end, all throughout the auditory system, up to the auditory cortex. And this is how we started out. When I went to NIH, as Dr. Danek mentioned, and then later at Georgetown, we took animals, rhesus monkeys, who had very similar brains as we do, just a little smaller, but their basic layout is very similar. And Brodmann actually did set architectonics on them as well. So visual cortex is back here, and auditory cortex is here along the lateral sulcus. And the unfortunate thing about the auditory cortex compared to the visual cortex is that auditory cortex is not as accessible for study. The visual cortex is easy to just remove a little bit of the skull in an anesthetized animal, and then you can study the neurons that sit there and process visual signals, whereas the auditory cortex is buried in that deep sulcus and it's hard to access. So we wanted to study auditory cortex that is out on the open surface here. And people had tried that before and didn't succeed because cells there don't respond to pure simple tones. They actually want complex tones. They wait for more interesting signals because it's already the secondary or tertiary cortex. So we used a very simple trick to get these neurons going. And just to briefly say, again, this was anesthetized monkeys that we studied with microelectrodes. So we open up the brain and the dura that covers the brain, and then you can see the brain surface with the blood vessels, and we can stick microelectrodes into different places and then determine the preference of single neurons in these regions to different frequencies, bandwidths, etc. So what we found is that neurons in these regions don't like pure tones of a single frequency so much. They respond much better when we make the sound broader. So like a kind of it's a noise, kind of a noise signal, but it has a certain pitch, and you can define them by varying the bandwidth of the signal, so you get different sounds there. Actually, I don't have an example of, unfortunately, but they sound fairly natural and commonly used. And these are the responses of the neurons in terms of their firing rate, or the firing in spikes per second. And we compare the firing rate to the noise signals to the firing rate to the pure tones, and you see that all of the neurons, basically all of them, with a few exceptions, respond better to these pitch noise signals than to the pure tones. And that was a sort of a first indication that as we go up from the primary auditory cortex to secondary and tertiary areas, there's more and more integration going on across frequencies. And that's what you want to see when you are interested in processing of complex sounds like speech. And then the next thing, so this is actually an additional result that we got out of these studies. Most of the neurons actually preferred a particular kind of noise with a particular bandwidth and frequency and so on. So these were interesting findings to start with, but they didn't tell us, of course, everything about what we wanted to know. So the next step was to use the language of the monkeys. We didn't want to use human speech because we didn't think that was something that the monkeys would be interested in. Although, of course, they hear us speak, but they're more interested in hearing their own fellows and their own mates talk. And there was a rich literature already at that time in the 1990s by people like Mark Hauser, who wrote the whole book about it, about communication systems in different animals, and he was an expert on monkey communication, and he gave us all these sounds that he had the digital library, and he knew exactly what these sounds meant. Some of them are just friendly contact calls that the monkeys use when they see each other and greet each other, basically, and then others are like words signifying particular food types, and they get excited and find particular types of grapes or other plants that they eat and want to tell others that they found this. So I'm going to play a few of these examples just for entertainment purposes, and you will notice that some of them are more friendly than others. So this is an arch scream for this is a very common one here. The coo in the middle in the top row. These are contact calls. It sounds friendly, doesn't it? You know, this is what they say when they see a friend, and they can actually vary those coo sounds a lot. So what is often said about monkey calls that they are sort of stereotyped and always the same, and it's not quite true. So Mark actually studied coos in a series of papers that he published. They are very adaptable, and they are, at least to some extent, learned. That they can express certain nuances about their in a social situation. The growls are, of course, a little more aggressive, and they probably fulfill the characterization of stay away from me as an aggressive call. But these are, if you just look at the spectrograms as phoneticians, you will agree with me that the elements are very similar. The melodies calls are harmonic calls. Some are noisy, but frequency changes are very common in there. So these are the information-bearing elements in a monkey communication calls are pretty much the same as in human speech. So we thought this is probably the right thing to do and study the effect of using these communication calls and record from neurons in the auditory cortex and see what these neurons do. That's what you would do as a human neuroscientist if you wanted to understand how speech sounds are coded in the brain. You would use human speech sounds and play them back, and this is actually what's happening now in epileptic patients. There's a program in San Francisco where a group is actually studying speech sounds, and we are in good communication with those colleagues, and they, I think, learned some things from us, and we are learning something from them. But I think it's generally, as a sort of off-the-cuff remark, I think it's important to have animal models that you can then compare later with findings in humans and see how they correspond. So what you can see here is that different types of monkey calls that are both very complex sounds, but about the same duration of maybe 200 milliseconds, evoke different responses. This is a typical single neuron response in a single neuron of the monkey lateral belt cortex, secondary auditory cortex. And the points, the dots that you see here are action potentials that the neuron fires in response to the sound. So the sound is presented here, the dark bar, horizontal bar that you see, and when you see an increased number of these dots, it means the cell fires, and then you can add those up. You repeat the same sound like maybe 20, 30 times, and then add them up. This is called a histogram. So you see the peak here, and this is a really nice response of that neuron to this particular sound. And here is a different sound, and the neuron doesn't like it that much. It also kind of notices it, but doesn't get crazy about it. And this is typically what we find. And now we can ask the next question. Why do particular neurons respond to particular communication calls and not to others? And so we're asking the question about neural mechanisms of, just like the people in the visual system asked, how do we see a face? How do we recognize a particular letter or other shape? So we have the monkey call here and play to the neuron, and the neuron responds with a beautiful response, a huge peak. This is kind of the noise of the neuron firing about when you don't give us a sound of any kind. And then what we do is we filter the call in different ways. So we take away the the low frequencies and only play the high frequencies out of this call, or we do the opposite. We only take the low frequency portion and take out the high frequency portion. In both of these cases, it has a pretty strong effect. There's no response now. As you can see, there's no peak like this to either the high or the low frequency. But you might say, why is that possible? Well, it's a nonlinear summation. So both of these frequency ranges have an influence on the response, but one alone is not enough. So they have to come together, and this is confirmed by putting the two pieces back together again, and the response is there like it was in the first place. So call this nonlinear spectral summation, and a summation in the spectral domain or frequency domain. Same thing happens in the time domain. So we have monkey calls that consist of different syllables, you might want to say, sort of like a word in spoken speech. And we get a nice response. And you might notice it's not at the very end. So the cell recognizes the call already after maybe the third syllable. It knows what's coming, that this is a grunt. But as we play just the first syllable or the first two syllables, that's not good enough. There's still no response. If we play the last three syllables, it's a tiny response, but not much. If we play it in reverse, there's also no response. So it has to come in the right order. And both of these properties, the spectral summation and the temporal summation, can be explained by a similar model, namely that the neuron that we're looking at gets input from different frequency domains, and it adds up all these inputs in a logical AND fashion. So all of them have to be present at the same time for the neuron to cross the threshold and to actually fire. So we end up with something we could call a bark detector, because this is a bark that I'm showing here. And time domain, we can now do more complex models. We have to introduce what's called delay lines. So the output of one set of inputs has to be held up a little bit until the input from the other set of inputs comes, and then they add up again, and then the neuron will fire only if the input comes in that particular sequence, but not in the reverse sequence. And so on. So one interesting observation that we've made is that monkey calls, as you saw, have a lot of harmonics, like human speech sounds have, or like music has that we like. And those neurons are there in the monkey as well. That neurons that prefer harmonic sounds over non-harmonic sounds. And it's, of course, an open question whether this is something that is innately there, or that is something that develops during experience. I had another teacher here, I was telling Wolfram the story earlier today, here in Munich, Terhard, who some of you might know, maybe not. He was sort of in the same department as Eberhard Zwicker, and he was studying speech processing and its development. And he was convinced that our preference for harmonic sounds comes from experience. He says, well, babies hear in the womb already their mothers speak, and the speech is harmonic mostly, so they develop this preference. And I wasn't so sure about this. I said, well, why couldn't it be an innate preference also for harmonic sounds that our auditory cortex has? And he gave me a bad grade, because I had a different opinion. But it's, I think it's still an open question whether this is something acquired or innate. People in the visual system are still arguing about whether the oriented bar detectors that they find there are innately determined or not. So I think it's, in the end, I was actually doing my doctoral thesis about this topic, so that's why I'm kind of traumatized about this, I think. But it's, in a way, it's a silly question, because it really doesn't get to the essence of what we want to study. It doesn't really matter whether this is innate or acquired, but the fact is that there are neurons that prefer harmonic sounds over non-harmonic sounds, and they play their role in the communication system. So that was sort of starting out from the very bottom, if you wish. Now we're getting to the next level of how does the brain put these things together. When you have these neurons that respond to particular sounds, you can imagine that the same thing happens in the human auditory cortex that we have neurons that respond to particular speech sounds, and then ultimately words maybe, and then how do we process sentences? Then it gets more complicated. So we are interested in this question now, most of all, and we took a model again from the visual system, which is well developed, where we've known for now maybe 20 years that there's not just one visual area, it's not just the visual cortex in the back of the brain, area 17, as Brodmann called it, but there are lots of associated areas that are working in parallel or in series to that. So down here's B1 in the monkey brain, down the temporal lobe here, there are all these areas that are shown in red here that contribute to object perception, and the face cells sit somewhere down here, and people have found neurons that respond to hands and to other kinds of complex visual scenes. And then there's another pathway, and these are anatomical pathways as well as functional pathways that go up into the parietal cortex that responds to well-localized visual stimuli. People, of course, with lesions in the parietal cortex get what's called spatial neglect. So they're not blind, but they ignore certain stimuli that occur in certain parts of the visual field. And these areas are also important for eye movements to a certain extent. So then they meet again all the way in the front of the brain, frontal cortex. That's where some kind of convergence happens, although this is really something we don't understand well, but it is well established in the visual system that there are these dual pathways that do different things, that do complementary things. And when we started to study the auditory cortex in the monkey, we began to also identify different regions, like what I told you earlier was done in these three regions, AL, ML, CL, as we call them, which are the next level from primary auditory cortex. So here's primary auditory cortex, area 41, as it was called by Brodmann, with its input from the thalamus. And these are areas that we and others defined in the last 20 years. The anatomy was known before that. There were studies by Ted Jones, by Pandya, various people in the United States who characterized, like Brodmann did, some of these regions on an anatomical basis, but we were among the first to study some of these regions in terms of single cell recordings in animal models. And what turned out to be the case is that the neurons that I showed you that are selective for complex sounds and could be used for something like speech processing sit mostly in these anterior regions. So area AL was particularly sensitive to the type of call that we used, whereas CL turned out to be more interested in the location where we presented the sound. So the monkey sat, and again, anesthetized monkeys in this silent chamber, with an electrode in the auditory cortex, and then presented the different sounds from different locations. I think I actually have a slide describing this here. So we had the monkey calls that we used for stimulations, and we had loudspeakers pretty much around the monkey in the frontal part of the room, and then tested both the type of call and the location of the call to see what the neurons preferred. And it turned out that, as I said, the monkey call preference, the type of call, was mostly processed in this anterior region, although there were some exceptions. There was some back here, but in the very back there was none of that. And conversely, in that part, which wasn't interested so much in the monkey call type, we had neurons that were very sharply tuned for the location of the call, whereas they were absent in the very anterior portion. So we developed our model on the basis of these findings, that there is also anatomically as well as physiologically a difference between the anterior regions and the posterior regions in the auditory cortex. And we thought that this could be the beginning of a dual pathway system like in the visual system. And to be sure that we were not sort of fooled by anything else, we also did the anatomy by putting anatomical tracers into each of these areas that we had identified physiologically, and then looked at the tracer, the labeled cells labeled by the different tracers in the frontal cortex. And sure enough, we found this is experiment that one does with an anatomical tracer. One first maps these areas with different frequencies, puts the tracer in which has different colors, and then you can later, you have to unfortunately kill the animal for that. Like, Boardman's work was done in dead people. The cells that are labeled are retrieved under a microscope. It's very laborious work. You don't want to do this too often. It's really hard work. And Liz Romansky was the postdoc in Pat Golden-Rakic's lab at Yale University who did most of the work, and she deserves all the credit for it. She found, after probably a whole year of analysis, really, that the anterior portion, AL, projects mostly to this ventral portion of the frontal cortex, and the posterior portion projects mostly to these more dorsal portions of the prefrontal cortex. And this is interesting because in the visual system, again, people found the same difference, that this region of the frontal cortex is more devoted to object perception, whereas the more dorsal part is more devoted to spatial perception. And so we found a very analogous organization of the auditory system into a ventral stream that is important for object recognition, if you define this in general terms, but also speech perception in our particular case, and a dorsal stream, which I will talk about in a minute, that is important for space perception, but also other things, and we'll get to that. So here, first of all, now we get into the realm of functional imaging in humans. And maybe some of you have been waiting for this and have been working on using similar methodologies. This is the best you can do so far in humans to characterize different regions of the brain functionally. So you have an MRI of the human brain here, which shows you the structural MRI, shows you the different part of the brain. You can see the temporal lobe here, and different depending on where you put your slice in, you can find auditory cortex quite nicely. And then we stimulate with sounds of different types, pure tones, bandpass noises, as we used in the monkey, and vowels as a sort of the simplest form of a speech sound. And we get the same kind of concentric organization that we found in the monkey, with a core area in the middle that responds readily to pure tones, surrounded by a belt that responds better to the bandpass noise, and then a vowel region that is mostly anterior of this so-called belt region. And so this is, first of all, was a good experiment to validate what we found in the monkey. And now we are sort of eager to translate some of this into the human. So one of the next experiments we did, and this was all done at Georgetown, of course, no longer NIH, but we're still collaborating with NIH. The next step was to actually use speech sounds. Like we used monkey communication calls in the monkey, we now use species-specific communication sounds that use speech in humans. And we also added other categories like musical instrument sounds. We also had bird song or other animal calls that we tested and looked at the activation in the auditory regions. And there is a region on the left side in particular of auditory cortex that very readily gets activated by speech sounds. So people have found this more and more. There are clusters of cells, probably, of voxels, as we call them in imaging, that cluster together where speech sounds are preferentially processed. There's another region that likes the musical instrument sounds. We did not find particularly specialized regions for animal calls. Why? Because it's probably not as important for us as speech is, or because the cells are more distributed, or because our method is not sensitive enough. But certainly speech sounds, if you were an alien and looked at these pictures, they would say, well, this must be a sort of a good sound that activates, at least a good portion of the neurons there. And they would be right. And so the idea of an anterior ventral stream as opposed to a posterior or dorsal stream that projects into the parietal cortex was tested by other people. Some of them set out to explicitly disprove our theory and said, well, we want to show that Rauschecker is wrong. But most of them came out with the same results. So there's a very prominent lab in Montreal at McGill University, Robert Satoru's lab, who didn't want to disprove us, but they did complementary experiments and came to very similar results, that the anterior part of superior temporal cortex is very good at discriminating different complex sounds, including speech sounds. And the posterior, this was a different lab in Cambridge, found that the posterior portion of auditory cortex projecting into inferior parietal cortex is very good at distinguishing different sounds at different positions in space and also motion in space. So the model of an anterior and posterior stream or ventral dorsal stream is now readily accepted and has been taken up by people specifically in the speech sciences and in linguistics, and has sort of taken off from there. One of the first people who specifically was interested in how speech sounds at different levels of complexity are processed was Jeff Binder, someone who agreed with us from the beginning. He showed that, well, different types of, different degrees of complexity within speech sounds activate regions anterior to the primary auditory cortex. So this is a horizontal slice through a human brain at different levels, and primary auditory cortex would be roughly here, and this is all in front of primary auditory cortex, so it agrees with our ideas that we developed on the basis of our monkey studies. And in particular, syllables then go readily into the prefrontal cortex as sort of something very essential for our speech understanding. And in our own lab, Jonas Oblaser, who came as a PhD student originally, or even master's student originally, and then came back as a postdoc, and Jonas just got his professorship in Lübeck a couple of months ago, with intermediate stations in London and Leipzig at the Max Planck. But Jonas did some of his first speech studies in my lab, and found that simple speech sounds like vowels, as we use them in some of our other studies, are better stimuli than bandpass noise, as you might expect in some of these speech areas, and they activate those anterior regions again that we first identified in the monkey and then in humans as well. And the remarkable thing about this finding, this was like almost 10 years ago, people were surprised by this, because when you go back to the textbooks, Wernicke's area is in the back. If you remember, I showed you that picture, Broca's area and Wernicke's area, and all the classical textbooks say, well, speech perception happens in the posterior superior temporal. So this was one of our problems that we had from the very beginning with the monkey findings, that they said, well, Wernicke's area is in the back, and you find your neurons in the front, you must be wrong, monkeys are not a good model for the human. And now suddenly, these imaging data come out, and they find most of the activation in the front and not in the back, where one would expect them according to the textbooks. So, long story short, this model is wrong. This is the classical textbook model where auditory information travels from primary auditory cortex into the back of the superior temporal, into the parietal cortex, and then through the arcuate fasciculus, it arrives in Broca's area, and from there you do speech production to the motor cortex. It misses an essential part, and that's this region, in front of the auditory cortex. And why that was never really in the old studies appreciated, I don't know, but even until recently, the anatomical connection from this part of the auditory cortex to the inferior frontal cortex was denied or was ignored. Von Cremon here in Munich was actually one of the first to describe this connection, and now it's well accepted. And it turns out that we were right after all. And that one of the breakthrough studies was this one, actually, done by a graduate student of mine, Ian DeWitt, who is actually dyslexic himself, has a light form of dyslexia. But what he did was a so-called meta-analysis. And you take all the published data in the field that were done with imaging in humans over the last 25 years that used different levels of speech, starting with phonemes to words and to simple phrases like hello, how are you, sort of just a few words, not long sentences. And then what he did is to take the coordinates, it's a good publication, they publish the exact coordinates in a normed database of X, Y, and Z in the brain and assign certain Brodmann areas to them. And so you can then put that into your own database and do what's called a meta-analysis. So you plot the group data from these, I don't remember how, several dozen studies, hundreds of patients or subjects that went into this. And what comes out is that phonemes are processed preferentially in a region anterior and lateral to primary auditory cortex, or Heschel's gyrus. Words are preferentially processed in even more anterior and lateral regions, and simple phrases are processed even more anteriorly. So there's your ventral stream right there, as we found it in the monkey. And Wernicke's area, if you define it as the area of the part of the brain that processes speech, which is, maybe you might argue with that, but that's one possible definition where speech is processed, that had to move from here, where it's described in most textbooks, to here. So we talk about a relocation of Wernicke's area. Now, the other possibility is, of course, to redefine Wernicke's area and say, okay, maybe we were wrong in the first place to say Wernicke's area is where speech is processed. Undeniably has something to do with speech and language, because people who have lesions there have problems with speech and language, but it may not be the coding of the speech sounds that happens there. Maybe something else happens there. And that something else is now the center of everybody's attention. So back to the monkey for a minute. How am I doing on time? 10 minutes. Okay. So I think, it's not my last monkey, that's right, but sort of a slight detour back. I told you about the connections that the anterior ventral pathway takes from primary auditory. Actually, best to start out with this slide, which was done by Chris Petkoff, who was a collaborator on this study as well. And this was done in Nikos Logatidis' lab, whose name you're probably all familiar by now. Went through all the papers. One of the most prominent monkey physiologists in the world, who was harassed by animal rights groups for years, and finally gave up to do this kind of work because he couldn't tolerate and bear it any longer. He got death threats, he got letters from people saying, tomorrow at 3 o'clock you'll be dead, and those people are still not in jail. So, we've lost one of our best non-human primate physiologists. But I published a paper with him just recently showing that the ventral stream is originating in primary auditory cortex and excites regions in the anterior superior temporal, and from there regions in the prefrontal cortex. And the way we showed this here was a different technique. It's called microstimulation, where while the animal is in the MRI scanner, you can stimulate parts of this region electrically with small electric currents, and that sort of like anatomical tracers gives you the locations that are connected with that region. Oops. So, a long story short, this is a sort of a complex study, and I'm only showing one slide here, but it again kind of makes the same point that there's information flow from primary auditory cortex to the anterior superior temporal and into prefrontal cortex, and that's pretty robust technology that we've used here. Now to the dorsal stream. The dorsal stream was originally described in the visual system as implementation of space processing. I told you that patients who have lesions in the parietal cortex, especially on the right side, often come up with deficits in space perception. They actually ignore or neglect everything that is present in the hemifield opposite to the lesion. So it's well established that those parietal regions, at least, that receive input from the posterior dorsal parts of the visual system as well as the dorsal part of the auditory system have something to do with space processing. That's undeniable. Also motion processing, to the extent that people in the visual system even at some point were thinking about establishing another pathway specifically for motion. That wasn't necessary. It was ultimately all subsumed into the space model. And now we're thinking, and there was also some evidence for that from the visual system, we think that the serial order of stimuli is also an important parameter for neurons in the dorsal system that kind of excites them. And the idea came from a music study. So in a lot of this that I've talked about and I've said about language, we could also bring up music as another example of complex sounds and of sounds that are organized in frequency and in time and that is sequence sensitive. And the study that I'm going to tell you about in a minute was really inspired by the Beatles. Because the title here, the heading, does not come from me. I had to give several interviews when the study came out, including Radio New Zealand and the National Public Radio in the United States. And the idea is very simple, and I'm sure you've come across this, most of you have. When you take, if you're old enough, that you grew up with the Beatles, which some of you are and some of you are not, then you might remember that there are these kind of what we called LPs, or then later there were CDs. All this has kind of disappeared. Now we have iTunes and MP4, whatever. But if you had an LP of the Beatles and you put them on your player and played them always again and again, like I did every day, the same record, and you play them always in the same sequence. There was no shuffling, like with the iTunes. And now you suddenly on the radio hear an old Beatles song, and you enjoy it, and you hear it at the end, something in your head plays the next Beatles song on your old LP. How does that happen? Same thing happens with classical music fans, of course. If you hear the end of the first movement of Beethoven's Fifth Symphony, at the end you hear the beginning of the second movement without anybody reminding you, and maybe you couldn't even do it. I could not. That's what I told one of the reporters. If you'd asked me what comes after Michelle on the White Album or whatever it is, I could not tell you. But if you play it to me, then I can continue. So the brain somehow remembers the sequence. And this is auditory memory, you kind of store speech sounds and music in a particular sequence. Otherwise, it is not that word or that sentence or that musical piece. It becomes that piece because the sounds appear in a certain sequence. So sequence is incredibly important for both music and language. And the results of that study, so two students actually, graduate students in the lab, Anne Lever and Brian Zielinski worked on this. And their joint data boils down to the fact that auditory memories like this, sequences, are stored in the motor system. And preferentially, auditory cortex is still important, I'm not denying that, but sequence information is all in the dorsal stream, or in part of the dorsal stream that is connected to the motor system, like this supplementary motor area here, which is the medial surface of the frontal cortex, or the dorsal premotor cortex, ventral premotor cortex. These are areas that light up when you image people while they're not hearing anything, while during the silent anticipation, after they heard the end of the first track, waiting for the next track, and the next track pops up, and we tested that behaviorally. We verified that, of course. That we had college students from Georgetown that we invited to the lab and said, bring your favorite CD. And then we tested them, could they sing the next track when we played one. And only somebody who tested at 90% correct got into the study. And in all these cases, during the silent anticipation of the next track, they activated their dorsal stream or motor cortex or premotor cortex. Some of them also activated their basal ganglia, their cerebellum, all motor structures. Of course, we've known for a long time that these so-called motor structures also have cognitive functions. And this is obviously one of them, that sequence processing is an important cognitive function, and it's accomplished by these regions. In this experiment too, Amber, who ended up being the first author on this study, and she's now a postdoc at UCLA, had the clever idea to actually train subjects with newly composed melodies. So she was a music major, and in addition to being an anthropologist and neuroscientist, she had the ability to compose little melodies and then had the subjects learn to associate each melody with another one, pairs of associated memory. And during the learning phase, the basal ganglia were incredibly active. They were important in chaining those sequences together. Once they became perfect, the frontal cortex took over, and it's what's called a clustering or I'm missing the word now, but patches of these neurons are being created that are then stored in memory. So this study is sort of still being debated a lot, and we're using new paradigms now to figure out which of these areas does what, because they all must be doing slightly different things in terms of sequence processing. But I'm convinced that this is what allows us to process sentences and complex memories. We're also studying conductors now who are learning new pieces, and as they are learning them, we are studying their dorsal stream to see what goes on there. I think I'm going to have to skip over some of these next slides. There's an MEG study there's a number of MEG studies that we were involved with Rita Hari's lab in Finland. Helsinki, I spent some years there. There's category, I know some of these data will be interesting for you if you're a linguist. Some of the categorical perception happens in the premotor cortex. So when you do morphing of different phonemes and then have subjects identify them without them knowing that they're actually phonemes, premotor cortex is the most active, in addition to some of the auditory areas, of course. And you've come up with this model that auditory cortex processes the spectral content, but then the temporal content and the order information is relayed to the premotor cortex. So this is your dorsal stream here that does some of this processing. And this is then the model that came out of all this, published with Sophie Scott a couple of years ago, where the green part is the ventral stream, where object identification happens. The red part is the dorsal stream together with parietal cortex, and premotor cortex forms the timing information, sequence information. And of course, there is information flowing back and forth between the motor system and the sensory, or in this case, auditory system. There are all kinds of terms in robotics that are being used to describe this sensory-motor interaction. It all boils down to sensory-motor processing, really. And terms, if you're familiar with the terms intrinsic, internal model, forward model, etc., this is where it comes into play. And we're getting more and more sophisticated describing the circuitry of these internal models, and trying to assign different roles to these different cortical regions. And if you invite me back next year, hopefully we'll know more about this. We are collaborating with some robotics colleagues here at the Technical University of Munich. Gordon Cheng is one of the collaborators, you might know. And I think this is a very promising way to actually figure out what computationally happens in the brain as we process complex information. So sort of the last just conclusion now is, are primate studies relevant for understanding language, or is it, should it all be done only in humans. Is there anything that primates have that would lead us on the right pathway? We think so. The circuitry is very similar, both anatomically and functionally. And just earlier this year, I published a paper with a bunch of linguists who are now in Australia. Ina Bornkessel was trained in Leipzig, had a professorship in Marburg. Her husband had a professorship in Mainz. They both left for Australia. And Steve Small is a language scientist at UC Irvine. And they are convinced that monkeys can tell us a lot about human language. They've used some of the same models to describe what happens in processing sentences, even at as high level as a discourse where people talk with each other, and use the terms that we've used for the monkeys to describe some of these phenomena in terms of predictive coding and internal models, error signals, etc. You have to read the paper. I won't be able to tell you everything that's in there. But I think it all shows that it's worth talking about these things, and we immediately got a response by somebody very prominent in the field saying, well, but you overlooked this and you overlooked that, maybe didn't cite them. But the problem is that the findings come up so fast and so quick that people who don't know about them overlook some of the most recent findings in the monkey literature, for example. And the claim that was made by some people that the difference between humans and monkeys is that they lack a direct connection, monkeys lack a direct connection from the posterior superior temporal cortex to the frontal cortex, the Broca's area, is no longer true. So we had a very easy answer for them and said, well, you're wrong. So this is the evidence here. This is diffusion tensor imaging in the human brain. There's the direct projection that I was talking about, often referred to as the arcuate fasciculus. People in linguistics have often claimed that this is what's missing in monkeys, and that's why monkeys don't have language, but the connection is actually there. So these are tracer studies that are even more reliable than DTI, and they clearly show there is a connection from here to here. So this cannot be the reason. There must be other reasons, and we think the reasons are in the frontal cortex, because the frontal cortex is the most advanced part in the human brain, and this is what we have to study in order to figure out why we have language as sophisticated as we do and monkeys don't. But I think the fact that they have at least rudimentary communication systems is very important, and we can use modern imaging techniques both in the monkey and in the human. And I think I should probably stop here and we'll skip the videos that I had planned to show, but I think that would take up too much time. So I just want to thank some of the collaborators. This is Mort Mishkin, who was responsible for bringing me to the United States, to NIH. Pat Goldman-Rakich, who passed away several years ago in a car accident. And Liz Romansky, her postdoc, who did all the anatomical work in the monkey. Piao Tian was my 12 years collaborator, who's now at NIH as a grants administrator. This is Mark Hauser, Chris Petkov, Nikos Logopidis, who we've collaborated with. Max Riesenhuber is a colleague at Georgetown, who I've been able to drag into auditory neuroscience more and more. He grew up at MIT. Well, grew up in Frankfurt, Germany, but came a postdoc at MIT before moving to our place. And these are the people who did the work in the monkeys: Pavel Kusmirek, Denis Archikov, Michael Ortiz, who's actually intubing at the Max Planck now with Logopidis, and Yuki Kikuchi, who's now in Newcastle, England. And these are the MRI people in the lab. Anne Maliba, who's now moved to UCLA, and various other people have left the lab. I mentioned Ian's work. He's at NIH. Laura and Brandon are currently doing their PhD with me. And, you know, I can never give enough credit to all those people who have helped with this. And the funders, of course, NIH, NSF, the Academy of Finland, and now I'm a senior fellow at the EIS in Garching, and so I'm hoping to do some interesting work there as well, in collaboration with maybe some of you. Thank you. Thank you.